Welcome to AI Dispatch, your channel for the latest AI videos on YouTube. If you don't want to miss these updates, please like, subscribe, and share. Core Memory Podcast brings us an interview with MIT neuroscientist Ed Boyden. His lab has developed a technique that sidesteps the expensive optics problem in microscopy by physically expanding brain tissue using polymer chemistry borrowed from diaper manufacturing. The approach converts nanoimaging from a hardware limitation into a sample preparation step. Boyden extends this logic further. He treats biological systems not as wet chemistry mysteries, but as mappable physics problems awaiting sufficient data resolution. We'll examine his case for why simulation, not dissection, will define the next era of neuroscience. Ed Boyden's lab at MIT has spent years solving a problem the microscopy industry assumed required better glass. For 3 centuries, neuroscience relied on improving lenses to see smaller structures. The diffraction limit of light created a hard ceiling, pushing researchers toward electron microscopes that cost millions and operate in vacuum chambers. Boyden's team rejected the lens improvement path. They asked whether the sample itself could be modified instead. The result is expansion microscopy. Brain tissue is infused with hydrogel monomers, the same polymer used in absorbent diapers. Adding water triggers swelling. The tissue expands uniformly at the molecular scale, stretching to many times its original size. A synapse that was previously too small to resolve with standard optics becomes visible under a modified webcam lens. This recasts nanoimaging from a scarcity problem into a chemistry problem. High-resolution brain mapping no longer requires institutional budgets. The constraint moves from expensive hardware to sample preparation protocols. Boyden refers to this as democratizing access to molecular scale imaging. A university without an electron microscope can now map neural circuits using equipment that costs less than a laptop. Boyden applies similar reasoning to the broader question of what biological systems are. Medicine typically treats the body as fundamentally opaque. We test drugs empirically because we lack complete knowledge of every molecular interaction. Boyden rejects this framing. He uses the term ground truth, borrowed from physics. In physics, we know the particles and forces. That knowledge allows us to build bridges that don't collapse and circuits that function predictably. Biology lacks that parts list. We don't know the precise location and state of every protein, every nucleic acid, every ion channel. Boyden's claim is that expansion microscopy and related techniques can generate that list. Once you have the molecular coordinates of a living system, you convert an organism into a dataset. That dataset obeys the laws of chemistry and physics. At that point, simulation becomes possible. He estimates that within 3 to 5 years, we could simulate a small brain or a single cell with full molecular fidelity. Within 10 to 15 years, a mammalian or human brain. This reframes the human body from a biological mystery into a debuggable system. Clinical trials could run in simulation rather than on patients because the simulation is grounded in physical law, not statistical approximation. If mapping the brain converts it into data, the next question is how to write data back into that system. Standard neuroscience uses electrodes to record activity or electrical stimulation to perturb it. Boyden compares this to fixing a computer by hitting it with a hammer. The tool lacks precision. His lab works on holographic optogenetics. Specific neurons are genetically modified to express light-sensitive proteins originally found in algae. A holographic projector then delivers 3-dimensional light patterns into the brain. Each neuron receives a targeted optical signal. This allows precise activation or inhibition of individual cells within a living network. The implication extends beyond research. If you can deliver arbitrary light patterns to specific neurons, you could theoretically encode information directly into neural circuits. Learning would no longer require sensory input over time. Information could be written onto the hardware. This raises the possibility of bypassing traditional education entirely, replacing it with direct neural programming. Boyden also addresses consciousness, a topic philosophers have debated for centuries. The hard problem of consciousness is the claim that subjective experience cannot be measured or explained by physical processes. Boyden reframes this as a data flow problem. Currently, we infer consciousness from behavior. A person speaks, moves, responds. We assume they are conscious. During a daydream, however, rich internal experience occurs without any external behavior. To an observer, nothing is happening. Boyden's position is that if we have a complete simulation of a brain, we can identify the circuit patterns that correspond to internal processing without motor output. Consciousness becomes a specific type of information flow that can be isolated and measured. The subjective barrier dissolves into observable neural dynamics. Boyden also offers a critique of the current artificial intelligence trajectory. The industry operates on the assumption that scaling compute and data will produce human-level reasoning. Larger models trained on more text will eventually surpass biological intelligence. Boyden points out that large language models still make errors no 5-year-old would make. Biological brains possess efficiency and creative mechanisms that silicon architectures lack. He highlights what he calls constructive failure. A human brain can daydream, form irrational connections, and stumble upon solutions through processes like dreams. The structure of the benzene ring was famously discovered through a dream of a snake eating its tail. Current AI systems are optimized to predict the next token, not to hallucinate novel realities that turn out to be correct. His position is that brute forcing intelligence with electricity misses the core mechanism. A human brain runs on roughly 20 watts. We are using millions of times more power to achieve inferior results. The path forward is not bigger GPUs. It is understanding the molecular wiring of biological systems. The future of AI depends on reverse engineering the brain, not outspending it. Boyden's work across expansion microscopy, optogenetics, simulation, and consciousness shares a methodological core. He refuses to accept complexity as an endpoint. Whether it is expanding tissue to avoid buying a better microscope or treating consciousness as a circuit pattern, he operates on the assumption that every phenomenon, including subjective experience, has a physical substrate that can be mapped and manipulated. The open question is whether society is prepared for a world where biology is no longer a mystery but a fully readable, editable, and simulatable system. If you're interested in the video featured in this episode, click the link below to watch the original.